Hello students, welcome back to my channel CSI Buddy. In today's video we are going to study about the Kreb cycle, which is the part of biochemistry. So in eukaryotic cells we have already studied about glycolysis. Glycolysis means converting one glucose molecule into pyruvate. And we got certain amount of ATP energy while doing this step. So the video for the same, that is glycolysis, I have already uploaded. So I will be providing the link in the description, so you can see that video if you haven't watched that. So in today's video we are going to learn about Kreb cycle. So once the glucose is converted into pyruvate, the pyruvate enters into mitochondria. But pyruvate won't enter the mitochondria as it is. It would first convert into acetyl coenzyme A, and then it will get into the Kreb cycle, which is going on in the mitochondria. So as shown in the figure, that the glucose to pyruvate synthesis, that was glycolysis, is held in cytoplasm. But after the pyruvate formation, the pyruvate is converted into acetyl coenzyme A, where coenzyme A is added to pyruvate, and then it enters into the Kreb cycle. So now we are going to study about Kreb cycle in detail. So as you can see in the figure, this is the channel which is present in the mitochondrial membrane, right? I haven't drawn the whole membrane, so you can figure it out. Now the pyruvate has entered and converted into acetyl coenzyme A. This is due to coenzyme A, which is an enzyme, that is PDH complex. PDH means pyruvate dehydrogenase complex. It is giving out CO2 molecule, that is pyruvate, which was a 3-carbon molecule, now is converted into 2-carbon molecule because one carbon is lost, and 2 NADPH are produced in this particular step. So in the first step we are getting CO2 out, right, and 2 NADPH is produced. Now this acetyl coenzyme A plus the OAA, that is oxalic acetate, would make citric acid, and the enzyme which is helping this formation is known as citrate synthase. So acetyl coenzyme A and oxalic acetate are together forming an citric acid. So as you can count the number of acetyl coenzyme A, that is 2-carbon molecule, and of oxalic acetate it is 4-carbon molecule. So citric acid would be a 6-carbon molecule, right? Now the citric acid is converting into isocitrate, the enzyme which is known as aconitase. Now isocitrate would convert into alpha ketoglutarate with the help of an enzyme isocitrate dehydrogenase. So as you can see, till this isocitrate the carbon number hasn't changed, that is 6 carbon. But in this case, when isocitrate is converting into alpha ketoglutarate, then 1 carbon is lost, that is the molecule now formed is a 5-carbon. So the CO2 has been lost. And whenever you see dehydrogenase, then it's obvious that a molecule known as NADH has been produced. So NAD positive would convert into NADH. So wherever you see the enzyme, that is dehydrogenase, then there would be production of either NADH or any other coenzyme which is having hydrogen, that is FeDH2. So now alpha ketoglutarate would convert into succinyl acid coenzyme A. So in this case we are adding a coenzyme A, so CoA would be added. The enzyme which is helping this process is known as alpha ketoglutarate dehydrogenase. Again you can see the term dehydrogenase, which would mean that this step also NADH positive NAD is converted into NADH, right? Now after this step we got succinyl acid coenzyme A. Now succinyl would convert into succinate with the help of an enzyme succinyl coenzyme A synthetase. Succinyl, now it would be releasing coenzyme A, and when the coenzyme A has been released, it is producing some kind of energy. So this energy would be used by the GDP plus inorganic phosphate to convert into a GTP molecule. Now again the ADP would be converting into ATP molecule, and this GTP would be converting into again GDP. So this is how the production of ATP is happening in this step. Now after succinate formation, the next step is the formation of a fumarate. So the enzyme involved in this is, and the enzyme which is helping it is known as succinate dehydrogenase, and this particular enzyme is present in the inner membrane of mitochondria. Right, so again dehydrogenase means that hydrogen is taken away. So in this step also FAD positive molecule would be converting into FADH2, right? In the next step fumarate is converting into malate, and the enzyme which is involved is known as fumarase. So in this particular step the H2O is lost. So now fumarate has converted into malate, and malate again converts into oxalic acetate, and the enzyme which is helping it is known as malate dehydrogenase. Again dehydrogenase, which means NAD positive would convert into NADH, right? So this was all about the Kreb cycle. Now going further, regulation of enzymes. Now how the enzymes are regulated. So the very first enzyme is the citrate synthase. Now citrate synthase has ATP. When ATP level is increased, so now we don't need more ATPs to be produced, so it would have an inhibitory effect on citrate synthase, and no more Kreb cycle would be regulated. Again, if there is higher NADH molecule and we don't know, we don't need anymore, so it would have a negative impact on citrate synthase. Again, if citrate molecule itself is very high in concentration, then also it would have a negative impact. Then succinyl coenzyme A, if it is in a higher level, so it would also have an inhibitory effect. But if there is a high amount of ADP, that means that low amount of ATP is present and we need more of ATP, so the citrate synthase would have a positive impact of higher ADP concentration. Now the regulation of isocitrate dehydrogenase, again ATP, the higher level of ATP would have a negative impact. Now Ca2 positive, that is calcium, would have a positive impact. Now calcium, as we know that it is required for the muscle contraction, and muscle contraction is directly related to ATP. So ATP provides the energy for the myosin to detach from the actin. So, and Ca2 positive is directly involved in the contraction of muscles, so it would have a positive impact, because after this Kreb cycle we get more and more of ATPs. Now ADP, high level of ADP would have a positive impact. Succinyl coenzyme A, again succinyl coenzyme A, if there is a high level of alpha ketodehydrogenase, it would have an negative impact on succinyl coenzyme A. Again, if there is more amount of NADH positive, so then also succinyl coenzyme A would have a negative impact. And again Ca2 positive, that is calcium, would have a positive impact on succinyl coenzyme A. So now we will see the gross products. So we are getting 4CO2. So this cycle is like pyruvate is two times. So we will get 2 times CO2. So this cycle technically is going through 2 times. So we will get 2 NADH positive. In this case also we will get 2. So we can calculate that in gross we are getting 4 CO2, 6 NADH and 2 FADH2 and 2 ATPs. So that was it about this video. I hope you enjoyed this lecture. If you like this video, please like, share and subscribe to my channel, and please help me to grow faster and faster. Thank you.